In this video, we will guide you through the lab procedure of the GA-map Dysbiosis Test Lx. For each step of the procedure, we will give a brief theoretical explanation followed by the practical lab instructions. Before initiating the lab procedure, there are a few notes that should be made. Appropriate personal protective equipment such as lab coat and disposable gloves should be worn to protect both the operator and the samples while working. Waste should be handled according to the local guidelines. All required instruments should be properly installed and qualified, and all equipment and reagents should be available and within expiration date. It is important that the instruction for use is read carefully. Refer to the instruction for use for details about additional reagents and required equipment, volume calculations, instrument settings and programs, storage conditions for samples and sample intermediates, and storage, handling and preparations of the different reagents. Let's have a look at the GA-map Dysbiosis Test Lx reagent kit. The kit consists of two boxes, box A, which is stored at -20 degrees Celsius, and box B, which is stored at 4 degrees Celsius. Box B also contains two buffers, which should be transferred to room temperature before use. The kit can be reused 3 times within the expiration date. Note that some of the reagents are single-use only and are provided in 3 aliquots. The lab procedure can be divided into 6 main steps. It is recommended to perform the steps in different rooms or zones to limit the risk of contamination. The first 2 steps should be performed in a pre-PCR room, while the last 4 steps should be performed in a post-PCR room. Preparation of master mixes should be performed in a clean, template-free room. Genomic DNA extraction is the initial step of the procedure. In this step, the bacterial cells in each sample are broken apart through mechanical and chemical lysis. When the cells are completely lysed, the DNA is isolated from the sample, washed and eluted by use of DNA-binding magnetic particles. For this step, you will need patient samples, lysing matrix E-tubes, and DNA isolation kit. Make sure the kit reagents are prepared according to the user manual. You will also need a fast prep bead beater instrument, water bath set to 65 degrees Celsius, and a Kingfisher Flex extraction robot with plates. The first steps of the analysis should be performed inside a biological safety cabinet. Working with 1 column at a time, unscrew the lids from the lysing matrix E-tubes. Vortex all samples prior to extraction. Add elution buffer to all tubes. And then add each sample. Refasten the lids, then repeat for all columns. When all the samples are transferred to lysing matrix E-tubes, process the samples twice in the FastPrep 96 bead beater. Transfer the racks to the plate centrifuge and spin down the samples. In the meantime, prepare the lysis plate. Prepare the lysing protease mix by adding lysis buffer and protease into an appropriate mixing tube. Pipette up and down until the mixture is visibly homogeneous. Do not vortex. Distribute the lysis protease mix to all wells of the deep well plate. Remember to include wells for negative controls. When the centrifugation step is completed, return to work in the biological safety cabinet. Work with one column at a time. Transfer the sample supernatant from the lysing matrix E tubes to the corresponding wells of the lysis plate. Mix well by pipetting. Lysis buffer is added as a negative extraction control. Cover the plate with adhesive PCR film and make sure the film is thoroughly sealed. Carefully place the lysis plate in the water bath at 65 degrees Celsius and incubate for 15 minutes. During this incubation time, prepare the buffer plates for the gDNA extraction according to instruction for use. When the 15 minutes incubation is completed, remove the lysis plate from the water bath. Carefully remove the PCR sealing tape and transfer the samples to plate number 1. Mix well by pipetting before and after. Turn on the DNA extraction robot. Initiate the GA-map program and follow the instructions to load all plates into the robot in the correct order. The procedure will take approximately 40 minutes. When the program is finished, remove plate number 5, which is the elution plate now containing isolated bacterial gDNA. Cover the plate with sealing tape and spin down in a plate centrifuge. Place the plate on ice and transfer the gDNA to a 96-well microtiter plate. Dilute the gDNA in nuclease-free water. This diluted gDNA is now ready for downstream analysis with the GA-map Dysbiosis Test Lx. Let's move on to the second step of the analysis, amplification of the 16S rRNA gene. All bacterial genomes contain the gene encoding 16S rRNA. This gene consists of regions that are highly conserved among all bacterial species, and other variable regions that are specific for certain types of bacteria. Specific primers will attach to the conserved region of the gene. With the help from a polymerase, a large part of the 16S rRNA gene will be copied. This reaction is repeated several times until thousands of gene copies are available in each sample. In the amplification step, you will need PCR master mix and kit controls, PCR polymerase, and PCR negative control, the same as used for gDNA dilution. You will also need a PCR plate with cap lids and a thermal cycler. Make a PCR mix by combining PCR master mix and polymerase. Mix by vortexing and briefly spin down. Distribute the mix into the applicable wells of a PCR plate. If relocating the plate to a different room, seal it with an adhesive plate seal. Controls and samples should now be added to the PCR plate. Mix well, both before and after transfer of the samples. Seal each column with cap strips. Extra mixing can be performed by vortexing the plate. Spin down the plate briefly and initiate the amplification program on the thermal cycler. The next step of the analysis is quantification of the PCR product. The quantification step is an important quality control checkpoint. It controls that the prior steps have been successful and verifies the quality of each individual sample. The recommended assays are specified in the instruction for use and will not be shown in this video. In the fourth step of the procedure, the PCR product is cleaned up. After the PCR, cleanup is important to degrade any primers and leftover dNTPs from the samples. The two enzymes involved in this process are exonuclease 1 and SAPE. They are added directly to the PCR product and are active during incubation at 37 degrees Celsius. After a short incubation at 80 degrees Celsius, the enzymes are inactivated and the PCR product will be ready for use in downstream analysis. In this step you will need exonuclease 1, SAPE, and a thermal cycler. Prepare the cleanup mix by combining exonuclease 1 and SAPE in equal volumes. Make sure the mix is homogeneous by pipetting up and down several times. Do not vortex. Transfer the cleanup mix to a reagent reservoir. Then add the correct volume of cleanup mix to each well. Mix well by pipetting, then vortex the plate. Spin down the plate briefly and initiate the cleanup program on the thermal cycler. The next step of the analysis is end labeling of the probe set. Recall from the amplification step that the PCR product now contains many copies of the 16S rRNA gene from many different bacteria, and that the gene copies contain variable regions that are specific for each bacterial type. In this step, the GA-map probe set is added to the PCR product. The probe set contains 48 different probes with complementary sequences to these variable regions of the gene. If a probe can find a complementary sequence, it will hybridize to it, and a probe will be labeled with a biotin molecule at its end. This is what we referred to as end labeling. Only the probes that find their complementary region among the gene copies will be biotinylated. For the end labeling step you will need end labeling master mix, biotin and end labeling polymerase, end labeling positive control, and end labeling negative control. You will also need a new PCR plate with lids and a thermal cycler. Prepare an end labeling mix by combining end labeling master mix, biotin and polymerase. Mix by vortexing and briefly spin down. Distribute the mix into each well of a new PCR plate, according to the plate layout. If relocating the plate to a different room, seal it with an adhesive plate seal. Controls and samples should now be added to the end labeling plate. Mix well by pipetting before and after. Seal each column with a cap strip. Extra mixing can be performed by vortexing the plate. Spin down the plate briefly and initiate the end labeling program on the thermal cycler. The last step is the hybridization and SAPE labeling. In this step, the end labeling product is added to a mix of magnetic beads. There are different types of magnetic beads, one for each probe in the probe set. On their surface, each bead type has a short specific oligo sequence, complementary to the probe sequence from the probe set. During the hybridization step, both biotinylated and unbiotinylated probes will hybridize to the beads. Following hybridization, the samples are incubated with SAPE, a fluorescent molecule that strongly binds to the biotin molecule of the biotinylated probes. This way, the fluorescence signal will correlate to the amount of target bacteria in the sample. In this step, you will need hybridization control from box A, water, bead set, and SAPE from box B, hybridization buffer and detection buffer. You will also need a magnetic ring plate and a hybridization plate with seal, and a thermal cycler. Make sure that the Luminex instrument is turned on and prepared for use for the final detection step. Work on room temperature during this step, but keep the end labeling plate and the hybridization control on ice throughout the procedure. Start by diluting the hybridization control in water. Prepare the hybridization bead mix by combining hybridization buffer, hybridization controlled dilution, and bead set. Mix by vortexing for 10 seconds. Do not spin down. Distribute the mix into each well of the hybridization plate, according to the plate layout. Add the controls and samples and remember to mix well by pipetting before and after. Seal the plate with a microseal A and initiate the hybridization program on the thermal cycler. During the hybridization step, prepare the reporter mix by combining SAPE and hybridization buffer. When the hybridization program is finished, add the reporter mix to the hybridization plate and mix well by pipetting. Reseal the plate with the new microseal A and let the plate incubate at 45 degrees Celsius for 30 minutes. When the program is finished, place the hybridization plate on the magnetic ring plate separator and let the beads collect on the side of the wells for 1 minute. It is important that the beads do not dry out during this wash step. Remove the hybridization and reporter solution carefully. Add detection buffer to all wells, then remove in one step. Remove the hybridization plate from the magnetic ring plate and add detection buffer. Mix well by pipetting until all beads are resuspended. Load the plate in the Luminex analyzer and initiate a batch with the GA-map protocol. When the run is completed, export the CSV file. This CSV file can now be used to generate results in the GA-map analyzer software.